Okay, we're ready to roll. Good afternoon, everyone. Thank you for tuning in. Just a few housekeeping notes to get out of the way first. First of all, please refer to the website that you're seeing on the screen right now if you'd like to see recordings of the seminars, and you can also see the schedule of upcoming seminars as well. Just a reminder to please post your questions into the Q&A tab, not the chat tab. Please post them in the Q&A tab, and I'll be able to look through them at the end of the talk, and then I can select some of the questions from there. So with that, it is an absolute pleasure and honor for me to introduce Dr. James DeGregory today, who is here from the University of Colorado School of Medicine. Dr. DeGregory is the professor of biochem and molecular genetics and immunology. He's also the Courtney C. and Lucy Patton Davis Endowed Chair in Lung Cancer Research. He is familiar with Boston. He did a stint there to get his PhD at MIT. I also want to note that he's the editor-in-chief of a brand new journal, Aging in Cancer. So you might want to check that out. Again, it's my pleasure to introduce Dr. DeGregory because I've been very familiar with his work that is really quite seminal in the area of cancer evolution and aging. His work has not only provided insights to cancer evolution, but really has shifted a paradigm in the way we think about cancer, not just a disease of mutations, but really how our body handles those mutant cells. So I'm really excited and looking forward to hear what he has to share with us today. So with that, Dr. DeGregory, thank you for accepting our invitation, and we look forward to your talk.
Great. Thanks so much, Sandy. It's really a pleasure to be here, and I really appreciate the invitation. I've been following Dr. McAllister's work as well, and she's a fellow traveler in aging and cancer. And an additional thanks to Nellie Polyak and David Weinstock for organizing this just wonderful series. It's really been a light in an otherwise at times dark year. And finally to Claudia Steele O'Sullivan for really organizing these talks and keeping all the logistics going. So with that, I'll move on. So, and it's not letting me advance. Oh, there it goes. So I have nothing to disclose. So the question we're really going to be tackling today is, why is it that 90% of cancers develop after the age of 50? And so I think we all can appreciate this in the sense that we know that, that we get this logarithmic increase in cancer incidence with life. And really about after the age of 50, your odds of getting cancer are substantially higher than before 50. But the real question is, why is this? And so to give a quick outline to my talk today, so we're going to be addressing this question, and first we're going to explore the current paradigm, and I'll discuss some of the problems with it. We'll talk about how evolutionary theory can inform on this question, and how some of a better understanding of sort of how organisms have evolved over billions of years can really help us to better understand the process of cancer. We'll then go into a theory or a model, maybe I shouldn't call it a theory quite yet, adaptive oncogenesis, that my lab's been working on for about 20 years at this point. And yes, there will be data. Some of it will be computational, although I won't really show much of that. Some of it will be in mouse models. And finally, we'll talk about some human data. And we'll also discuss, or I hope you'll appreciate, why all of this matters for strategies to avoid the scourge of cancer. So from the conventional paradigm, the reason we get cancers as we get older is due to the time and cell divisions, the exposures we receive, and our underlying genetics. And all of these are supposed to really impact one parameter, and that's mutations. So almost all models of the age dependence of cancer have focused on mutations, and that's been basically the parameter of interest. And this has led to the current paradigm that really posits, in fact, it's the current paradigm for probably over half of a century, that cancer incidence is limited by the occurrence of oncogenic mutations. So the reason we get this logarithmic increase in cancer late in life is due to the stepwise accumulation of oncogenic mutations. And you need to develop sort of a full house of these driver mutations before you can develop a cancer. So, but if we step back and we think about, immortality, we know that humans have longed for immortality for many, many centuries. And so is cancer going to stand in the way? So is this sort of, inevitable accumulation of mutations that happens throughout our life going to really prevent us from achieving this goal? And in addition, a lot of people think that it's actually the accumulation of mutations that causes aging in general. So is the solution to really avoid mutations? So if we think about this, how does evolution work? So now we're talking evolution of organisms. So we do know, so first off, is evolution driven by the occurrence of the right mutations over time? And of course, the answer would be no. Evolution has been driven by environmental changes. So over the last 700 million years or so, at least for, animal evolution, we know that there's been dramatic changes in the species that inhabit our Earth. And often these periods, which, basically separate huge differences in the species that are present, have been punctuated by massive environmental changes, such as the meteor that hit off the Yucatan 65 million years ago. Thanks to this meteor, we're able to have this conference. And volcanic activity, huge changes in temperature and atmospheric gases. And these changes all drive selection for new phenotypes and new species that are adaptive to these new landscapes. And so this is how we understand the evolution of species. So should we be thinking about cancer in any different terms? So in this light, we can ask, are patterns of cancer incidence during a lifetime best explained by the sequential accumulation of oncogenic mutations, the conventional paradigm? Or do we need to consider cell extrinsic factors and evolutionary pressures? So to answer this question, a former postdoc in the lab, Andre Roshak, basically started by examining the cancer incidence curves from SEER data throughout life. And what you can see from this is, so for example, CML and AML and CLL have an age dependence, and they're driven by what we know to be different numbers of mutations, really one for most diagnoses of CML and multiple for AML and CLL. And you can also consider solid cancers, everything from colorectal through mesothelioma. And they too are driven by different numbers of drivers. And so these numbers are derived from Martin Carena's paper that basically calculated the expected number of drivers. And depending on whether you only look at known drivers or whether you look at all drivers, at least defined by sort of selection signatures, it's anywhere from 4 to 11 for colorectal down to about 1 for mesothelioma. And while these numbers are all a bit soft because they don't include, for example, epigenetic changes that could be drivers. They don't necessarily include large structural rearrangements that could be drivers. But I think it's safe to say that the number of drivers varies enormously across cancers, probably by about a log. And on top of that, we noted that these different cancers and leukemias initiate in very different stem cell pools. So for leukemias, they're initiating in many cases within hematopoietic stem cell pools on the order of 100,000 cells. And for colorectal cancer, they would be initiating within these small crypts, each containing probably no more than a dozen stem cells, but many millions of them. So huge difference in the pools. And yet despite this, when Andre did a very simple thing, he simply normalized the incidences of these cancers to their maximums. And when you do that, you'll see that they all fall right on top of each other. The age dependence is just strikingly similar. So again, this is despite the fact that some of these, like CML, are initiating in one type of pool and require only one driver, and others like colorectal cancer require many drivers and are initiating it in much larger pools, but basically that are physically separated into small pools. You still get this common curve. And even clonal metapoiesis, which we can be pretty confident in good measure is initiated by single variants, also shows a very similar pattern. So when Andre did computational modeling of this using Monte Carlo methods, first, when he tried to model it using the conventional paradigm, he basically couldn't find realistic parameters that would allow for such large variances in driver numbers and pool size and so forth, and still give you relatively similar curves of incidence. But when he included a very simple parameter, basically differential selection, such that in youth, the first half of life, that you had selection against driver mutations, and that late in life, this would be converted to selection for those driver mutations. So basically a conversion throughout life from negative to positive selection. That would allow for a much better approximation of these different curves. And I think this also helps us address what's been a paradox in the field since Richard Peto proposed this in the 70s, and this is why it's known as Peto's paradox. And that's how is it that organisms, in this case mammals, that range from the size of a blue whale to a mouse, which is a variance of about 10 to the 7th fold in terms of cell number, and probably a couple of logs in terms of lifespan. How does the blue whale and the mouse similarly avoid cancer through their reproductive lifespans? Certainly through their natural reproductive lifespans. And this is a real problem from the conventional paradigm because obviously it takes a lot more cell divisions and thus probably more mutations to make a whale and to maintain a whale. But from this other perspective, where it's simply this differential selection, as long as one maintains the negative selection for longer within the whale, it's less dependent on the numbers of cells. So Andre further proposed a model that could help explain this commonality of these incidence curves. And first off, let's hypothetically consider if there's an organism that could have a single somatic mutation, and this mutation would lead to malignancy that would reduce overall reproductive success because since it only required one event, it was going to be more likely to happen, and thus it would happen early. And another malignancy may require 2 events, and thus it's going to be of lower probability. And that might happen later, and thus have less of an impact on reproductive success. If this were the case, you would expect that over evolutionary time, you're going to get germline selection for cellular mechanisms that suppress somatic evolution. And this is going to depend on the effect of the particular mutation has on overall reproductive success, which is, of course, the currency of fitness. And so there'll be greater selection against this red mutation. In other words, selection for mechanisms, somatic mechanisms, to prevent this from leading to a malignancy. And you'll still get selection against the blue and the yellow mutations, but lesser. And you'll get some sort of equalization of these curves, essentially a tuning depending on how much they would affect overall reproductive success. So essentially, natural selection is pushing off the risk of cancers to the right side of this curve. And I'll note that this isn't unique to cancer. This is the case with heart disease, kidney failure. You pick your favorite malady and they're pushed off to the right. And of course, there's a reason for that. And that's because these diseases are occurring late in life because there would otherwise be minimal selection against these diseases beyond the age where most individuals would already be dead by other causes. So if we look at a curve for European survival about 15,000 years ago, and you could imagine this curve would be pretty similar for other peoples any thousands of years ago, probably anywhere on the planet, and that with every passing year, there was some odds that you weren't going to make it to the next year, largely because of extrinsic threats, from disease to hunger to predation, and such that there were some individuals that would make it, even to their 60s or so, but it wasn't as common back then, much less common back then than it is today. And you compare that to modern incidences of these diseases, and you can see that there's relatively little overlap. So that's not to say that aging and cancer and so forth were never a problem, but their impact on the fitness of these individuals, their ability to reproduce, was probably relatively minor. And that's because natural selection acted to push these to the right. They acted to develop methods or mechanisms to prevent these diseases from affecting fitness. And so you might imagine, well, why not push them further to the right? Because there were some individuals affected. Well, that would probably come with a cost. And that cost might be, for example, in reduced reproductive success. And thus, natural selection has balanced these and essentially allows for pushing it to the right, but not such that it never is going to affect an individual. So you might be thinking, well, yeah, but what about childhood cancers? Testicular cancers, which can often happen in men in their 20s and lymphomas in young people. Well, I won't have time to go into detail, but I'll propose that exceptions to this late life pattern will often be rare, associated with modern lifestyles in developed nations, and are associated with pathogens. And in each case, these reduce the power of natural selection to mitigate these effects. So if something is rare, the ability of natural selection to prevent it from happening is relatively weak, for example. So for these reasons, life is not linear. So up until even our mid-30s, the chance of dying, for example, from about maturity through the mid-30s in any given year is about constant. Your chance of dying in your 20th year is about the same as your chance of dying in your 30th year. And yet we know 10 years has gone by, 10 years of damage accumulation. And you know that you're 10 years closer to the end of your life on average when you are 30 than when you were 20. So, but despite that, it's relatively flat. And yet then we go past 40 and even into 50, and you could see that we start going downhill. Downhill in the respect that our odds of dying in the next year is greater than in the preceding year. And we don't just go downhill, we pick up speed. And this is despite the fact that this nonlinear pattern is despite the fact that much of what we know about damage accumulation, including mutation accumulation, is relatively linear from maturity on. And I think this really speaks to evolved mechanisms of resiliency, where what I like to call the 3 R's, redundancy, replacement, and repair, which I'll describe more later, are allowing sort of a pushing up of this curve to maintain us through those years of likely reproductive success. So now, getting back to cancer, what does this investment in tissue maintenance during youth and the waning of this investment in old age have to do with cancer? And this is the model that my lab's been developing for 15 to 20 years at this point, adaptive oncogenesis. And so this model really has two kind of key features. The first is that we propose that high stem cell fitness opposes somatic evolution and thus promotes the status quo. And so just like an animal species or bacteria or anything that have evolved within a relatively constant environment, they will evolve to be at some peak of fitness for any trait. So for example, swallows, if you think of swallows, longer wings would be bad and shorter wings will be bad. And so swallow wing length is maintained within a relatively narrow spectra to basically maximize fitness. And we propose the same thing is true for stem cells within our tissues. That at this somatic level, we have evolved a fit between our stem cells and their niches such that change in any parameter, any phenotype, will likely reduce the fitness of that cell. Now we're talking somatic fitness. And so reductions in the fitness will then lead to an increased chance that cell is going to be eliminated from the pool. So even if there was a malignant genotype over here that had a higher fitness peak, if getting there means going through low fitness intermediates, it's going to mean that this transition is going to be difficult and rare. And I use those words on purpose because it doesn't mean that a 30-year-old who does everything right can't get cancer. We just know that he or she is much less likely to get cancer than an elderly person or someone who smoked or anything like that. And so basically, the evolved type of a stem cell in the evolved environment that it finds itself in is going to be at some sort of fitness peak and thus resistant to change. But alas, we don't stay young forever, or we do things like we smoke cigarettes and so forth. And this will change that landscape. If you could think about the lungs of a smoker, for example, you can appreciate that a stem cell in that lung of a smoker will be in an environment that it did not evolve to be within. Not to mention that the stem cell itself could have accumulated damage over the years and epigenetic changes. So it'll be a poor match for its environment at this point. And this thus allows for selection for adaptive phenotypes that provide an advantage within this new environment. Just like the species that inhabited Earth adapted to the new environment after that meteor hit off the Yucatan 65 million years ago. Change promotes selection for new phenotypes. And while this doesn't always lead to malignant genotypes, it increases the odds that it can lead to a malignant genotype. So just to summarize that, we know, of course, from work of evolutionary biologists for many, many decades, the natural selection has acted over millions of years to maintain animal bodies to the extent that maximizes reproductive success. We can all accept that. And what I've told you is that maintaining tissues in their youthful state is potently tumor suppressive. So youth is intrinsically tumor suppressive. But the depressing part is that aging and tissue damage select for change, which can sometimes lead to cancers. So then what is the evidence for differential selection for somatic mutations depending on context? And so I'll first describe some work that was done by Curtis Henry when he was a postdoc and an instructor in my lab. He left about 4 years ago, and now he's an assistant professor at Emory. And what Curtis wanted to ask was, does the microenvironment of the bone marrow influence whether or not an oncogenic driver can be advantageous? And so he took young stem cells, selected out the stem and progenitor cells from the bone marrow of these young mice, put in an oncogenic mutation, in this case it was an NRAS activating mutation, together with GFP, and he purposely only put it into a small fraction of the progenitor cells. He then put this into busulfan conditioned mice, and that was another key feature, because if we had used irradiated mice, we probably would have seen expansion of these, because irradiation itself is very disruptive to the microenvironment. But he worked out a protocol that was very mild, that only caused transient inflammation, and that importantly largely left most of the mature cells intact. So it was a relatively minimal effect on the microenvironment. And he went in either young mice, old mice at about 20 months old, or siblings of these old mice that were IL-37 transgenic. So these transgenic mice, IL-37 is in the IL-1 family, but it actually suppresses IL-1 dependent inflammation. And so the important thing is the identical pool of cells with the identical oncogenically initiated cells is going into different microenvironments. And then when he asked, well, did these cause expansion? Strikingly, in the young background, he got about the percent that he put in. In other words, he didn't see any selection for the oncogenic variant within the young background. And this kind of goes against the conventional paradigm where the oncogenic mutation confers sort of an automatic fitness benefit. It did not. But in the old background, he gets these expansions. And it's variable. I'll note that everything we do with aging is variable. It's one of the hard parts about working in age models, but we do consistently see this expansion. Now, if he looked in the recipients that were old but that were anti-inflammatory, he largely suppressed these expansions. He then asked, well, what's underlying this? So we know that RAS can induce changes in signaling. So we looked at signaling. And so these are the young cells, and what we would say is this is the evolved level of phospho-STAT5 signaling. But in the old background, that's reduced, and it's very consistently reduced, and others have seen this too. But importantly, the cells that also express the NRAS, but what otherwise were old, or even if they were young in the old background, they basically restored this signaling. And note that the IL-3 and the IL-37 transgenic background, so the cells themselves don't express the IL-37 transgene, but the cells that were put in that would otherwise be suppressed come close to restoring their signaling features. He also looked at different gene expression events, and he noted that the cells in the old background have a lot of defects in sort of what we would think of as fitness parameters. So for example, nucleotide synthesis genes, MYC. These are all reduced within the aged backgrounds. And they're largely restored by NRAS expression. But you'll note that the expression of the IL-37 transgene in the background prevents that reduction from happening in the first place to variable extents. It doesn't prevent the GMPS much, but it partially prevents the MYC as well. So there's a partial restoration of the fitness of these progenitor cells when they're in the anti-inflammatory background. So I want to give a nod to the many labs that have shown similar things. And obviously I can't go into all of them, but you could see that, for different context of exposures and there's different oncogenic events that have been shown to be differentially selected for depending on these contexts. And of course, now we're getting a lot of this happening just from the deep sequencing of normal tissues that's happening. And there's many, many examples of this in human tissues. And some of them are shown here. They've shown in the skin, the esophagus, the endometrium, that basically you could see an age-dependent increase in these driver-driven clonal expansions. And sometimes not just age-dependent, so they can also be driven by things like colitis. So this leads to the model. And the model is that we may accumulate genetic damage throughout our lives. In fact, much of it's going to be front-loaded because we would expect the rate of accumulation to be greater during ontogeny when there's more cell divisions happening. And in fact, some of the recent data has shown that for the hematopoietic system. And yet, despite this genetic damage, which certainly is going to include oncogenic events, there's stabilizing selection. So there's the evolved type is the favored type, such that you're basically pushing down on the chances that these lead to a clonal expansion because they're at peaks of fitness, or at least local peaks. But late in life, when the force of natural selection to maintain our tissues has declined, and on top of that, because of accumulation of damage and so forth that's happened because of exposures we've received, now that same negative selection for those same events can be converted to more of a positive selection, where you could select for these clonal expansions, thus increasing the odds that there's going to be somatic evolution down a malignant path. So I'll now turn to another example. And this is work by a very talented MD PhD student in the lab, Kelly Higa. And she was co-mentored by myself and Eric Pietras, a faculty member at my university as well. And so this has been a very close collaboration. And basically, what they want to know is, are inflammatory cytokines sufficient to promote selection for oncogenic events in early hematopoietic progenitors? So one thing we know is that a number of different inflammatory contexts, whether induced by aging, radiation, or chemotherapy, have pretty consistent effects. If you look at a lot of gene expression data, they tend to reduce self-renewal of stem cells and at the same time promote differentiation, which are kind of two sides of the same coin. And in addition, Kelly noted that a number of different oncogenic events have a common feature as well, and that they promote self-renewal while inhibiting differentiation. And she also noted that many of these have negative effects, suppressive effects on CBP alpha dependent transcription. So they devised a model, or together we devised a model where we are using IL-1 beta as the inflammatory stimulus and loss of CBP alpha as the differentiation inhibiting event that would basically be a model for many different oncogenic events that have this as a common feature. And so what Kelly did is Kelly used mice that were engineered to either be mutant for CBP alpha or wild type, which specifically within the hematopoietic stem cell compartment. So she could induce this deletion, harvest those cells, and then she would create chimeric mice where only a small fraction of the cells were either the wild type or the delta genotype. She again used this busulfan conditioning to avoid the inflammatory effects of radiation. And then she treated for about 20 days with either PBS or IL-1 beta. And notably, you'll see that basically in the absence of the inflammatory stimulus in just PBS treated mice, the loss of CEBP alpha did not provide an advantage. And this actually went against previous publications, but those publications had used a radiation to condition, and they'd also used poly-IC as the way of inducing deletion. And as you all know, poly-IC is itself inflammatory. But when we treat it with IL-1 beta, you see a marked selection for CBP alpha loss. And this is seen in multiple stem and progenitor type populations, what I'll be referring to just as HSPCs. So then the question is, well, how does CBP alpha knockout provide HSPCs with a fitness advantage? And so Kelly performed RNA-seq on these sorted progenitor populations, and we're focusing on the MPP3 because that's where she saw the most dramatic selection. And we're going to be comparing at first all these orange cells, either the wild types or the mutant cells, either in the vehicle-treated background or in the IL-1 treated background. Now, the first thing you'll notice is that IL-1 itself induces massive gene expression changes within this progenitor population. But what was really striking and actually surprising is that loss of CBP alpha had a really dramatic effect at reversing these changes, and indicating that a substantial portion of IL-1 dependent transcriptional changes is CBP alpha dependent. We then want to know, well, what are those changes? And so she noted by first comparing the wild type cells with or without IL-1 beta, you see that there's a big decrease in the stem cell gene expression signature. And this is similar to work that Eric Pietras had done before when he was in Mano Passage's lab, and actually in his own lab as well. But what they then noted is that when you compare the mutant cells to the wild type cells in the presence of IL-1 beta, you could see that you could largely restore this gene expression pattern. So CBP alpha loss restores the stemness genes. On the other side, they looked at myeloid specific genes, and you see this massive induction of myeloid specific genes by IL-1 beta. And again, a really nice reversal of that gene expression changes when you lose CBP alpha. So CBP alpha loss reverses the precocious myeloid differentiation that results from chronic IL-1 beta treatment. And you can see this in vitro as well, which is a nice model system. So if we culture these MPP3 HSPCs in vitro with or with IL-1 beta, you see that IL-1, as expected, induced loss of the HSPCs. And that loss was largely restored, in fact, completely restored by later time points by loss of CBP alpha. So the reductions in the stem cells was restored, and that's consistent with that restoration of self-renewal genes. At the same time, if we look at the myeloid population, the mature myeloid population, you can see this precocious differentiation induced by IL-1 beta, and that is largely reversed by the loss of CBP alpha. So again, CBP alpha is preventing HSPC exhaustion and precocious differentiation. So if we think about this, we know that IL-1, largely through CBP alpha, is promoting myeloid differentiation and preventing self-renewal. This is important because it leads to increased granulopoiesis and allows one to deal with an infection at the expense of reduced HSPC fitness. But that if you remove CBP alpha, you reverse both of these effects, thus selecting for CBP alpha loss. Now, what was interesting is that Kelly also sequenced all the competitors. So these are all should just be identical BLAC6, MPP3, hematopoietic stem and progenitor cells. And the only difference is either what they're treated with, what the mice are treated with, or who they're competing against. And what I'll just show you now is these competitors that are either competing against the CBP alpha deleted cells or the wild type cells, but all in the presence of IL-1 beta. And what you can see is in the cells that were competing against the mutant progenitors, they have even more increases in myeloid gene expression and even more decreases in the stemness phenotype. So essentially, these guys are even worse off than these guys from a competitive sense because they're differentiating even more and they maintain stemness even less. And that's really cool. So it indicates that not only do these guys have a competitive advantage because they maintain those signatures, but they further bash the competitiveness of their competitors. So what I've told you is that IL-1 impairs stemness and promotes myeloid differentiation, all of which is reversed by CBP alpha loss. And so CBP alpha knockout confers a fitness advantage to these HSPCs, specifically in the context of IL-1. And we can understand how this, because of IL-1's causing this HSC dysfunction, it may serve as a driving force for this clonal evolution, which is shown down here. And so if we step back and we think about this from the evolutionary perspective, I think it makes a lot of sense. So what I've described is that we've shown that CBP alpha dependent program is required for emergence of granulopoiesis in response to an inflammatory insult. So this is going to be critically important for surviving infections. Big fitness benefit, right? Because these affect you at all ages. On the other hand, chronic inflammation can promote selection for oncogenic events that impair CBP alpha function. And this seems to be mostly the case given that when these leukemias occur in old age, and maybe in rarer cases before old age. So from an evolutionary perspective, it's a relatively minor fitness cost. And I think this harkens back to some of the antagonistic pleiotropy theory that George Williams developed back in the 50s. And he stated that natural selection may be said to be biased in favor of youth over old age whenever a conflict of interest arises. And I would say this is such a conflict of interest, but it probably wasn't a very tough choice for natural selection. So now we're going to turn to the final part, which is why does aging cause lung cancers? And this is the work of several talented students and postdocs in the lab. And so we'll start by just a simple observation that, no surprise to anyone, whether you get lung cancer is largely determined by smoking. So this is current smoker lung cancer, never smoker lung cancers for men and for women. You basically massively increase your risk of lung cancer by smoking. But what's really striking is when these authors did a simple thing and just did a log scale, you can see that these curves are quite parallel. And so even though whether you get lung cancer is determined by whether you smoke or not, when you get it is determined by your age. And so the age dependence of these curves, which is better seen over here on the log scale, are pretty much similar. The only difference is their amplitude. And to me, this really says that even when someone does something that's such a pro-carcinogenic condition like smoking, that youth is still powerful in suppressing these cancers. So we want to understand, well, what are some of the changes that happen in lungs as we get older? So we know there are lots of immune changes, declining adaptive immunity, a lot of inflammatory changes. There are a lot of functional and structural changes as well, where your lungs don't work as well. And so we address this using an experimental system that had been developed by the Ventura lab. And what they used is they created this adenovirus that delivers a couple CRISPR constructs, and one makes a break within an intron of the ALK gene and another within the intron of the EML4 gene. And this creates EML4 ALK fusions by basically an inversion of the chromosome that really mirror those that happen in humans. And I should mention that these fusions, these EML4 ALK fusions, are most common in non-smoker associated lung cancers. And so when we introduce these in, first thing we noted is that using phospho-STAT3 as a proxy, which the Ventura lab had shown gets induced by these inversions, you can see that the rate of inducing these is pretty similar in young and old mice in the lungs, looking very quickly after introducing the virus. But when we ask, what about the age of the host? And I'll note that we're using a dose of the virus that's about 10-fold lower than has previously been used, because we wanted to be able to see differential effects depending on ages. And so in the young background, note that each dash is a mouse. We get very few adenomas forming. Same thing in the middle-aged. But in the old background, pretty much every mice develops adenomas, and they develop lots of them, and they develop big ones. So essentially, we're getting more of these EML4 ALK-driven expansions leading to adenomas in old lungs relative to young. We also see that there's an increase in immune aggregates in these old lungs relative to young lungs. And Kathy's done a pretty comprehensive evaluation of the different cell types in here, but I won't be presenting that today. But there's a pretty massive increase relative to young. And then we want to know, well, could we reverse that? And so we used a transgenic model, alpha-1 antitrypsin, or what I'll be referring to as AAT. And we had previously shown, when this was work from Curtis Henry again, that this increase in inflammation that you get throughout the body gets decreased in old AAT transgenic mice in the bone marrow and the serum. And we also see this increase in these immune lesions in old lungs gets decreased as well by expression of the AAT. AAT is being expressed from the SBC promoter in the lung. It's secreted, so it goes systemically. But it's known to block neutrophilic protease reactions that initiate a lot of the inflammatory reactions. And so when Kathy is compared old versus young, she sees increases in the lungs of an inflammatory response. And again, this is largely reversed by the AAT transgenic expression. So just like I showed you before with the the systemic inflammatory cytokines, we see those inflammatory changes in the lung and how they get reversed. And so she then asked, okay, as before we showed, there's not a lot of adenomas being formed in the young mice. Most mice don't get any. You get very few of them. And the old mice, we get bigger adenomas and we get more of them. But what was striking in the old mice that are also AAT transgenic, we get essentially a youthful profile of adenomas, and that they're relatively rare and they're not that big. They then ask, okay, what about if you blocked adaptive immunity? Maybe it's the loss of adaptive immunity as these mice get old that's leading to this increase in adenomas. And they found that that doesn't seem to be the case, because there wasn't much of an increase in adenoma formation in young RAG mutant mice. That doesn't say that these cells aren't contributing somehow late in life, but it says at least loss of adaptive immunity in youth is not sufficient. So then we want to know, okay, so what's going on in the lungs of these mice? And these are mice that that don't have tumors. These are just basically young, old, and old AAT mice. And so we asked, what are the changes in proteomic profiles in these mice? And you'll see that there's a lot of changes that don't get reversed by AAT expression. And that's not surprising. We're not completely rejuvenating these mice. On the other hand, there is a subset of changes, probably roughly a third, that do seem to get reversed. So these are increases in old lungs that largely get prevented in the old AAT mice. And so what are, well, first, so one of the lessons I think from this is that we apparently don't need to completely rejuvenate a tissue to avoid oncogenesis. We don't necessarily need to find that fountain of youth. We just need to figure out what the key aging dependent changes are, and then figure out how to modulate these factors safely. That's not a small task. And so these are some of the changes that we observed. So we see big increases in S100A8 and A9, which are known to be pro-inflammatory. And those are mostly reduced by the AAT expression, which is shown here in blue. So gray is old, white is young, by the way. And you can also see that in the old mice, we get these big increases in some of the proteases, like the matrix metalloproteases and elastase, that are known to be involved in tissue remodeling. And those, again, are largely reversed in the old AAT backgrounds. And in a collaboration with Etienne Danis, we also did GSEA analysis. And so they used the GTEx data set, which we took young people and we grouped those between 20 and 39, and the older people, they were from 60 to 79. And they looked at basically gene set enrichment analysis for those RNA-seq data. And essentially what you see is something very similar to what we see in those old mouse lungs. You get increases in a bunch of inflammatory pathways, including the senescence-associated secretory phenotype pathway, the SAS. And at the same time, we see that you get these increases in ECM and tissue remodeling pathways, just like we're seeing in the old mouse lungs. So to just summarize this part, we've shown that EML4-ALK initiated lung cells preferentially develop into adenomas in aged backgrounds, and that old age is associated with increased inflammation and tissue remodeling in the lungs of mice and humans. And finally, that reductions in the inflammatory environment associated with old age can markedly decrease adenoma formation. So this fits because we know that aspirin and more recently from the CANTOS trial, anti-IL-1 beta treatments can reduce cancer risk in humans. But I'll note that we need to be quite careful. So for example, even in the CANTOS trial, they had a 3-fold reduction in lung cancers in people that were treated with anti-IL-1 beta, but no effect on overall survival. Mortality was the same because they had increases in deaths from sepsis. So we need to come up with means that are more targeted and less disruptive and important pathways like inflammation in order to really affect a good reduction that has less risk. It's also known that AAT deficient humans exhibit a several-fold increase in lung cancer risk. So if we think about this, some of the conventional paradigm, the causes like aging and smoking, they're causing cancer by causing mutations, leading to malignant phenotypes. And then when people think about the microenvironmental changes, or many people that are induced in cancers, for example, they see them as the consequence of the malignant phenotypes being induced by the mutations. And that is true to some extent. On the other hand, what we're focusing on is how these same causes affect microenvironmental changes, which then alter selection for adaptive mutations. And so we're focused on a different aspect of this, but I want to emphasize these are not mutually exclusive. So it's not to say that the malignant phenotypes themselves are not changing the microenvironment. In fact, I'd argue that there's a bit of a handoff happening. That early periods of oncogenesis are really dominated by this bottom part, but as the cancer grows, probably the normal tissue environment is less, sort of meaningful in terms of the subsequent evolution, and that it becomes more driven by the malignant phenotypes themselves. And intra-tumor competition becomes more critical relative to competition with some of the normal cells. So then this really begs the question of, how are tissues maintained in the first place? So I've talked about how tissue maintenance and that maintenance of a normal microenvironment can impede cancer. And I think it's important to step back and think about the many different mechanisms for this. And obviously, there's the cell intrinsic stuff like DNA repair and telomeres, but we also have a lot of redundancy. So we have redundancy in our stem cell compartments. If you have to eliminate a stem cell, you got thousands of them, it's really perhaps no big deal. There's cell intrinsic quality control from, apoptosis that's induced either because a cell's damaged or because it might be oncogenically initiated. There's immune surveillance. And there's cell competition. And this is really cool and beautiful work over the years from Drosophila has really revealed this beautifully. But where a cell that is either poorly functioning, or more recently, they've shown it even hyperfunctioning, meaning potentially oncogenically transformed, can be either eaten or pushed out by its neighbors. And on top of that, really great work from, over the years from Beatrice Mintz, Mina Bissell, and others has shown how peer pressure, normal structures favoring normal phenotypes, can allow for even oncogenically initiated cells, if they're within this normal environment, will basically remain well behaved. And I think this is really coming to be to the forefront with these recent studies of normal driver mutations found in normal tissues that appear to be still normally functioning. So all of this is involved in maintaining the youthful soma. So why does it all matter? Well, my lab is obviously focused on how this affects anti-cancer defenses. But I'd say this really is much more general, because maintaining a youthful soma is good for avoiding other diseases like heart disease, for maintaining good immune defense, for good tissue function, and of course for robustness. And all of this affects organismal fitness, because it's going to increase the odds that you survive and reproduce through those years when that was likely. And I don't think I can give a talk during this period without at least mentioning COVID-19. And so we've talked about this age dependence of cancers shown here in the colored bars. And you could see the same age dependence for different cardiovascular disorders and for other events as well. And you could compare these curves to the curve of basically fatalities among patients with COVID-19. And I hope you can appreciate these curves are pretty much the same. Again, after 50 is when these curves start increasing logarithmically. So some of the take-home messages are that efforts to prevent and treat cancer should really converge with similar efforts to prevent other aging-associated diseases. And we need means to shore up our bodies, the soma, to improve defenses against malignant actors. And we need to leverage markers of the declining soma, like clonal hematopoiesis, which has been beautifully shown by Ben Ebert and other labs to highly associate with cardiovascular disease risk and overall mortality. And others have even shown it associates with frailty. But basically, these are ecosystem checks for our soma. And so I've already thanked a lot of the members of my lab as we've gone along, and some of the former members. I'll mention, if you like the artwork, that's all by my son, Michael. And most of it's lifted from my book. We've had great funding from the NCI, NIA, and some of the lung cancer research was supported by the Cancer League of Colorado. Great collaborators at the university. And Dan Tenen for providing the CBP alpha disrupted mice. And I also want to thank Niles Eldridge. So I mentioned him on one of the earlier slides as well, but he's been a great colleague and friend over the last decade or so. And we've had many great discussions about, some of his theory and how it's very relevant. Some of the punctuated equilibrium theory that he developed with Stephen Jay Gould is very relevant to these cancer models. And there's the lab. Of course, we're zooming it. And finally, that was given permission to give a quick plug for the Aging and Cancer Journal, which you already heard about from Sandy at the beginning. And I'm happy to take any questions.
Okay, James, thank you. That was wonderful. Very thought-provoking. Gives us a lot to chew on. I'm glad you mentioned the book, because now I can say to everyone, if you want some holiday reading, get the book. It's special in many ways, in addition to the fact that, as James already mentioned, his son Michael did the illustration. So with that, I think we should get to some questions. There are a number of questions, and I think I'll start if I could.
Sandy, should I stop sharing, or what should I do with that?
I don't know.
Please keep your screen up.
Yeah,
Yeah,
okay.
Keep it like that. Thanks.
Okay.
Okay, great. So in thinking about CBP alpha story, certainly loss of CBP alpha was important for differentiation, stem cell renewal. What is the source of IL-1 beta? That one would normally say,
Right.
You have exogenous IL-1 beta, but would there be a source of IL-1 beta that would, keep depleting the stem cell pool and pushing that differentiation toward myeloid lineage?
Exactly. So IL-1 beta goes up during infections. It goes up in old age, after we get irradiated. And I'll note that one of the things that I didn't have time to present that Kelly recently did is she asked, well, what happens if you take away the IL-1 beta? And I'll note that this is one of those great examples where reviewers ask something and you do it and you go, damn, that was a good idea. And it worked beautifully. Basically, the selection goes away completely. So it goes back down to baseline. And so this sort of emphasizes that, the evolved function is going to be largely safe. If you get an infection, if you get something that's temporary, you might get that brief period where you would select for CEBP alpha loss. But as soon as the insult goes away and IL-1 levels go down, so does that selection. And IL-1, we've seen it with radiation, even post-radiation, it might last for a couple of weeks. It doesn't have long-term persistence. And I don't know if it ever goes down to complete basal levels after radiation, but it definitely goes back down. And same thing in old age. I mean, of course, in something like old age, it's going to be chronic forever. Unless you had an intervention, once IL-1 starts going up when you're older, it's going to stay up. And so that probably is the context that's most likely going to select for these events and thus allow for additional events to occur.
Oh, yeah, makes sense. So under those conditions, is there also, is there skewing of differentiation away from lymphoid population so that, you're skewing toward myeloid, but what's happening to lymphoid? Is that also getting depleted under those conditions?
That's a great point. Yeah. And indeed, all of those contexts are indeed associated with myeloid bias. So it's long been observed by Irv Weissman, and I'm probably forgetting many of the labs that have shown this as well, but where old age is associated with myeloid skewing. So hematopoiesis becomes much more myeloid as we get older. Same thing, after radiation exposure. And of course, all of this is an evolved response of protection, because if you get damage and if you get an infection, you need those frontline defenders, your myeloid cells, not only to clear infections, but they're also the front line for repair. And I think that's great in the short term, and it's obviously been great for us evolutionarily, but it can come back and bite us in the butt when we get old.
Yeah, so it makes me think too then, you mentioned COVID. So those conditions seem to me like it could explain, the decreased response to vaccine with age as well.
Right.
Are there CBP alpha mutations or loss of CBP alpha that I wonder, identified as, determining response to COVID and the vaccine.
Oh, it's a great idea, and I have no idea what the answer to that. I actually don't know if how polymorphic CEBP alpha is. I've never looked into that, but that's an interesting thing to look into, not only for sort of immune responses, can you better maintain more of a, some of your B and T lymphopoiesis, but I think probably the reductions in B and T lymphopoiesis that happen throughout a life. First off, some of that's programmed. I mean, a lot of that happens when you're still a teenager. That's not really aging dependent. It's in some ways by design. And I always think that, we may travel around a lot and get new exposures, but probably for most of our evolutionary history, you probably had experienced most things by the time you were 20 or 30. In other words, the idea that you're going to get a new pathogen probably wasn't that great. And so, we probably evolved to make these T and B cell repertoires, but then that was down-regulated at later phases of life because you already had created this army, and it was probably better to invest in myelopoiesis at that point. So some of it could be programmed, but I think this does get accentuated late in life. And the extent to which that is contributing to poor vaccine responses, I don't really know. Certainly the reductions in the production of new T and B cells is going to contribute to that.
Sure. Cool. So they're also sort of thought-provoking, thought questions too with respect to tissue maintenance. You mentioned youthful tissues can sort of tolerate mutations, but that decreases with age. So if I can combine a couple questions here about, suggestions for tissue maintenance, there's also an example provided by one of the questions that you may be familiar with this IMAGE, immunological age work that was put out of Technion. There was a paper published in Nature Medicine in 2019 describing, essentially looking at peripheral blood cells and coming up with a gene signature that correlated with immunological age and function, not necessarily with chronological age, but
Right.
in immuno age. So I guess it sounds like the question that's being put forward here is, could you envision something similar with tissues? Would we have a way of understanding what youthful healthy tissues are, and could we do anything about it?
Yeah, no, I think some of these methods for looking at epigenetic age and for people have come up with proteomic parameters, essentially that distinguish your chronological age from your biological age. And so essentially you can determine someone's age, but at the same time, you could say, okay, you're 5 years older than older biologically than your chronological age says you are, or maybe you're, great and you're fit and so forth, and you're 5 years younger. And so there are a lot of metrics of that. And I think in some ways those are readouts on our tissue, even if we don't understand fully how to read them out. I think I wouldn't be surprised if we could use some sort of circulating, cell-free DNA that circulates as sort of measures of tissue health. And I'm actually working with someone who is trying to do that. Though I think those are great questions in terms of maintaining tissues. I mean, in some ways we already know how to do it. You eat a healthy diet, you exercise, and you don't smoke. And, things like, for example, exercise, we know what it does. It ties into the autophagy pathway, which is a self-renewal pathway. And if you look at model organisms, flies, worms, and it's even been done in mice, that, if you select for longevity, whatever mechanism you do, like if it's through the IGF-1 pathway or anything, it almost always seems to impinge on autophagy. And I think in many of those cases, they've shown that those interventions, whether genetic or even dietary, like caloric restriction, they all require this boost in autophagy. So I would say if we could figure out how to manipulate things like autophagy, but not too much because you don't want to self-eat yourself away, then I think those are going to be the safest and most effective means. But really the safest and most effective means are things like good diet and exercise.
Yeah, cool. I'll be interested to see what you find in with some of those studies. We have a question from Dr. Milos Basic, who's a postdoc. Even though cancer risk increases with age, some cancers are more aggressive if developed in younger patients. Pardon me. Similarly, therapeutic responses can be quite different based on age. How does this impact your model and ideas of fitness, tissue maintenance, and age suppressive programs?
No, it's very true. There are definitely, like, for example, breast cancers that occur in women postpartum, which is sort of a whole different class of breast cancers, can have worse outcomes. And, I think people are still trying to figure out why that is. I mean, certainly your odds of developing breast cancer increase as you get older. And the same thing with lung cancers. Lung cancers that develop in younger people are known to be highly aggressive. And honestly, I could only really hand wave why that is. I mean, it could be that, like I said, natural selection is not perfect. It doesn't completely prevent every cancer from happening. And so if you do get such a cancer, maybe it had to sort of be super aggressive to be able to have evolved on a relatively normal landscape. And that's complete hand waving, but that's about all I got.
Yeah, I mean, I think that's interesting. To think of it in that sense, it's making me think of breast cancer, which is something we study, in that, the breast tissue develops later in life. I mean, late meaning during puberty. That's not, I wouldn't consider that old, but is there something to that? Tissues that are still evolving or developing, later in life,
Yeah.
again,
Yeah.
but the idea that there's that postpartum risk of breast cancer within a 5-year window. I'm thinking
Right.
your colleague Pepper Shadeen has done.
Exactly. So Pepper, Tracy Lyons. I'm actually working with Tracy Lyons on a project with this, and Ginger Borges, and they've been trying to address this question. And it seems that tissue remodeling that occurs, basically during involution has a lot to do with it. But one of the things that they've noted from the risk is that this is most prevalent in women that have their first baby after the age of 30. And it's barely present at all if a woman starts having babies, at 20, for example. And you can imagine that, again, natural selection hasn't had time to prepare for the modern world. And so natural selection selected for mechanisms that women would avoid cancer through reproductive years. But with the old way of doing things, where probably a woman, first off, was pregnant probably early in her life, went through far fewer menses because she'd breastfeed for 3 years and so forth, right? So I think we have to start to appreciate how these changes in lifestyles have influenced cancer risk and most likely even the aggressiveness of those cancers that develop, which I think is where we know much less about that.
Yeah. Well, unfortunately, we've reached our time. This was phenomenal. Again, thank you so much for accepting the invitation and presenting your work and giving us a lot to think about. I hope you and everyone here has an amazing holiday season. Stay safe, and there's so much hope ahead of us in the next couple of months.
Yeah. And I'll mention that anyone who didn't get I will get the list of questions, but feel free to email me or tweet me. Tweet at me, I guess is the better way to put it. And thanks so much, Sandy. It's a real pleasure and a real honor to give this talk in this series. Happy holidays, everyone.